Good day, everyone. I'm Butcher Evangelista from Pinnacle Medical. And first of all, we would like to thank our partners from the Philippine Society of Pathologists for letting our company partake in this 69th virtual annual convention. And we would like to thank all our viewers today and our guests. Before we begin and before we introduce our speaker, I would first like to introduce our company so that more or less you would know what we are about. And if you have any questions, you can also visit us in our booth, or you may contact us through our details here or visit our website. Our company, Pinnacle Medical, aims to be the leader in infection control products by 2030, and this will be to the benefit of our Filipino people through our healthcare services, our people, product, and programs. And our key advantage would be we work hand in hand with our affiliate, Evangelista Medical Specialty Hospital, and they act as a test laboratory to make sure that the functionality and the quality of the products which we distribute are properly assessed and evaluated by skilled healthcare professionals. Furthermore, we also have a quality assurance committee, and their role is to ensure that the products and devices which we distribute are relevant and responsive to the standards of patient care. Our clientele ranges from level 1 hospitals to tertiary level hospitals nationwide in both the private and government sector. We carry different brands as well, from infection control products such as Winner Medical to imaging principals such as Siemens, Fujifilm, Gold Care, and cold chain storage providers such as Biobase. If you would visit our website, you would also see that we carry PPE products, which is crucial today due to the pandemic, from N95 masks, surgical masks, and coveralls, which are all FDA approved here in the country. In terms of infection control in the operating room, we carry surgical drapes and infection control products and devices for different procedures. Advanced disinfection filtration systems for COVID-19, which can be used in almost all the departments in the clinics and hospitals. Now, with the entrance of the vaccines, we would also like to mention that we also carry cold chain products such as medical refrigerators and ultra low freezers for different types and brands of vaccines. For more details, you can also check our website or the booth. And finally, imaging products and devices should you be interested. Our key principle today would be Vazyme Biotech. So Vazyme Biotech is devoted to the development and production of enzymes and antibodies. One of their key products, which they are actually introducing in today's market due to the pandemic, would be the anti-SARS-CoV-2 neutralizing antibody Elasta kit, which is a rapid and high throughput evaluation of the efficacy of the vaccines. And I would like to play their company video so that you would know more about Vazyme Biotech.
In a few large antivirus battles in human history, when virus persecuted us silently, we didn't surrender. However, to defeat the invisible killer in human body, we need to know it. Only by knowing it as soon as possible can we obtain more time to conquer it, prevent more misfortunes from happening. The progress of human science shortens the time to discover virus. From over 5 months to discover SARS in 2003, to over 1 month for H7N9 in 2013, even now to only 5 days for the whole genome sequencing of SARS-CoV-2 with NGS technology. The second-generation NGS technology, mNGS, is used for SARS-CoV-2 sequencing. Isolation, cultivation, and purification of microorganisms are no longer needed. Nucleic acid could be extracted directly from clinical samples to build the virus library and get genetic information of pathogen from samples via NGS. Then, virus sequence, the picture of the virus, can be attained with bioinformatic analyses. The picture will be helpful to capture the virus. 3 ways to capture the virus are nucleic acid detection, antibody detection, and antigen detection. An all-round approach, leaving no place for virus to hide. The most rigorous and accurate method for nucleic acid detection is to extract nucleic acid from samples of secretion in respiratory tract, take the conserved sequences in viral genomes as targets, and design the corresponding primer and probe. According to the real-time quantitative fluorescence PCR technology, the target sequence index increases, and fluorescent signals appear after each round of amplification, during which fluorescent signals are accumulated and detected by relevant PCR instruments, while samples not infected by the virus will not show any PCR reaction due to the absence of relevant target sequence. Whether the sample contains the virus can be judged through fluorescent signals. The other 2 ways for detection are antibody and antigen detection. After initial infection of COVID-19, B cells are primarily activated to generate specific antibodies against the virus. Antibody detection demonstrates health conditions of patients through the detection of their specific antibodies. IgM, first antibody generated, remains in early and middle stage of disease onset and cannot be detected after body recovered for a period of time. IgG can be detected about a week after disease onset and used for diagnosis and tracing back infection history. The antigen detection targets at COVID-19 viral antigens. Its genes encodes multiple structural proteins, like N protein and S protein, with several antigenic epitopes. Because of binding between specific antigen and antibody, we can identify the existence of SARS-CoV-2 antigens, directly demonstrating if the sample contains antigens for COVID-19 virus. These methods in different methodologies vary in testing conditions, operational standards, and individual immunity. After knowing characteristics of the virus through detection, we take countermeasures. Our immune system identifies pathogens after viral infection, creates specific cellular and humoral immune protection against COVID-19 virus to remove pathogens, and generates immune memory cells to prevent secondary infection. In vitro building of system for immune recognition, protection, and memory against the pathogen in recovered individuals by simulation is helpful for the development of immunotherapy drugs and vaccine. Specific protective antibodies or antiserums developed against the COVID virus are effective for treatment under different scenarios. Vaccine carrying protective epitope information of pathogens recognized by immune system can simulate the process that pathogens stimulate immune system for specific protective immune responses and for immune memory. Attacked by the virus again, immune memory cells with protective epitopes will be rapidly activated to protect body and prevent viral intrusion and replication. All researchers are working against time for vaccine development. Evaluations on safety and efficacy are the core in clinical trials of vaccine. In Phase 1 and 2 clinical trials, the focus is on the dosage and procedure for immunization. Except monitoring and adverse reactions for vaccination, we should detect humoral immunity and cellular immunity to assess the immunogenicity of vaccine, indirectly reflect its efficacy. Large-scale Phase 3 clinical trials will be conducted in badly hit areas. Vaccine efficacy will be evaluated by comparing infection rate of vaccine group and placebo group. As time for R&D of COVID vaccine is greatly shortened, vaccine efficacy and persistence should be tested continuously after its launching. Since the formation of the universe, there have been countless battles between humans and virus. With strong desire for survival and life extension, virus constantly evolved and mutated. In such ceaseless war, human technology is advancing simultaneously, accelerating technological innovation, and achieving significant technological process in life safety and biosafety. Developing technologies to improve human health.
Our key speaker today is a college graduate and a postgraduate of Nanjing Agricultural University in China. He also has a joint PhD program in Germany, and he specializes in microbiology and immunology. Currently, he is also the R&D manager for Nanjing Vasai Medical Technologies. I would like to introduce Dr. Ke Ma.
Hello everyone, my name is Kema and I'm from the VZM company in China, and it's my great opportunity and I'm very honored to have this chance to give a presentation here. And today my topic is neutralizing antibody testing with high throughput and open system sVNT. So as we all know, the SARS-CoV-2, the COVID-19 pandemic has already lasted for almost 2 years around the world. So these figures show us a very simple structure of how the SARS-CoV-2 virus looks like. And clearly, this virus includes four major structural proteins: a spike protein, membrane protein, envelope protein, and the nucleocapsid proteins. These four proteins is very important for the functional or the infection for the virus, especially the spike protein. And in the human bodies, especially in the respiratory area, there is a lot of different kind of cell that express the ACE2 on the surface. The S protein of the SARS-CoV-2 virus contains a receptor binding domain, or short for RBD, and it is responsible for recognizing and interacting with the cell surface receptor, and that is the ACE2. So this is the structure of these two proteins we can see here actually. These two proteins can form a very strong bind interaction between each other. And so what's the meaning, or how the ACE2 and RBD binding helps the virus infect the human cells? So following the binding of the RBD to the receptor ACE2, actually the SARS-CoV-2 virus, the membrane structure will change and fuse it together with the target cell or the human cell, and lead to the release of the viral nucleic capsid into the cytosol of the infected cells, and the virus will start replicates in our cell and get ready express and explode for the next infection. So during the infection, our immune system, especially the human immune system, is also plays a very important role to help us and protect us to fight against the virus infection. So after receiving the antigen stimulation, the B cell from the naive B cell will activate it, and they begin to a series of proliferation or differentiation to form a plasma B cell, which can produce the antibodies, and the antibodies can help us to better fight against the virus. So here we show the kinetics, and almost based on the structure and the function, the antibodies in our body can be divided into the IgM, IgA, IgG, and IgE. And usually the IgE is more related to the allergies, and the IgM, IgA, and IgG plays very important role in our immune system to fight against the different diseases. So the figure here shows the natural infection of the SARS-CoV-2 virus and how the antibody changes from the very beginning to the long term. So as here we can see at the very beginning after infection around 10 days, the IgM and IgA, the level is increased very fast. So the IgM and IgA can provide the very fast and rapid protection to fight against the virus. However, the half-life of the IgM is very short. So after like 14 days after the infection, the IgM, the level is start to decrease. And for the long-term immunity, the IgG is the most important one. So usually after around 10 days after infection, the IgG started to increase in the circulation system, and we'll get into the peak after around 3 to 4 weeks after infection, and that will keep existed in our human bodies and provide human a long-term immunity. However, for the different diseases, the half-life of the IgG also different. For the SARS-CoV-2 virus, we still not know very clearly, so we have to put a big question mark here. But I think it's very important to continually check the IgG levels changing, especially for the convalescent samples or the people after vaccination. It can help us better prevent the diseases and to also to cure the diseases. So how the antibody protect our body, or how the antibody fight against the virus? So based on the function, actually the antibody can be divided to neutralizing antibody or non-neutralizing antibody. The neutralizing antibody persists in the circulatory system, could bind to the pathogen in the bloodstream. Partially of that could block cellular infiltration and replication and protect our from the superinfections or reinfections. Based on the current published papers, the neutralizing antibody against the SARS-CoV-2 is bind with the RBD protein. So after the neutralizing antibody bind with the RBD, the SARS-CoV-2 virus cannot bind with the ACE2 anymore, so the virus cannot get into human cell anymore. So we can block the virus infection at the very beginning. However, there's also some non-neutralizing antibody that combine with other protein on the virus surface. And for those proteins, if this antibody can bind the virus, but the antibody binding can still not block the virus binding with the ACE2, and the virus can still get into human cell. And the worstly is there will be some antibodies. After they bind with the virus, the Fc fragment can be recognized by the monocytal or the macrophages Fc receptor, which mediate the antibody mediate the inflammation, or the antibody mediate the infection, which make the patient's symptom even more worse, and can be developed to very severe diseases. So the classic method to analyze the neutralizing antibody is to use the live virus neutralization test, or the VNT. The VNT requires handling live wild-type or the mutant SARS-CoV-2 virus in a specialized biosafety level 3 or BL3 lab, contaminated facility, and in the laboratory intense and time-consuming. Usually that will take at least 3 days, but usually that will take 4 to 6 days to finish all the analysis. So for the live virus neutralizing test, because the highly requirements for the facility and the time consuming is not very easy to be achieved in all the lab around the world. And the substitute way is we use the pseudovirus-based virus neutralization test. Actually, for this method, usually we use like the HIV-based pseudovirus. We put the spike protein on the pseudovirus, and we also have the cells which can express the ACE2 receptor. So because the pseudovirus encodes the luciferase gene, so this the quantity that get into the cell model can be detected based on the luciferase signal. So for the pseudovirus, compared to the live virus one, it's much more safety because it can be done in the BSL2 lab, but because still based on the cell model, so it's still time consuming compared to the live virus one. The time costing almost the same. It also need 3 to 4 days to finish all the experiments. So now based on the situation, we want to develop a faster and much more easier to use the tool to help us to better understand or better detect the neutralizing antibody. So based on this, we developed the neutralizing antibody Elasta test. So the mechanism is really similar to the pseudovirus or the live virus one. We express the ACE2 protein and coated them on the Elasta place. And for the reasons, we have the HRP conjugated RBD. And so when we mix the HRP RBD together with the serum samples, if there is some neutralizing antibody that will bind with the RBD. So the RBD will not attached to the place anymore. So after washing, the signal can be checked. If more free RBD bind on the place, the signal will be higher. But if a lot of RBD were neutralized by the antibodies, the signal will be very low. So based on the signal, we can detect the level of the neutralizing antibody in the different samples. So here is a summary of the advantages and disadvantages between these three methods. So for sure, the VNT or the live virus neutralization test is the golden standard for study the neutralizing antibody. But compared to the pseudovirus experiments or the ELISA, it requires the BSL-3 lab, and it needs to operate the live virus. So, and it's very dangerous to be done in many lab or cannot be done there. So, and the duration for the experiments, the live virus analysis and the pseudovirus is quite similar because it's all based on the cell model. So 2 to 4 days is required. And the Elasta test, which can be done in the BSL-2 lab, which can be done in most hospital lab. And because there is no virus involved in this experiment, so it's the most safety experiments compared to the previous two. And for the Elasta, the test will be reduced from 2 to 4 days to 1 to 2 hours. So why the neutralizing antibody detection is so important? I think the most important function or most important objective to detect the neutralizing antibody is for the epidemiological investigation. So according to the WHO, it mentioned if their sample is positive or suspicious to be either the positive, neutralizing antibody analysis should be ideally be performed, especially with the now more and more people will get vaccination. So we want to know after the vaccination how the antibodies already created or not, or how the antibody changes with the time after the vaccination, how the antibody will change. We want to know the kinetics. When we should get the results, then we can help the better protection by the vaccines. So here is the components of the case and the figure. So all the components in the case will be those regions and one dilution place and one Elasta place which coded the ACE2 protein. And here is the CE certificate we already get and some validate, like in Indonesia we already done. So here is the manual protocol of how this process can be done. So because the Elasta method can be done manually, and we can see here, so the whole process can be finished within 2 hours. So within 2 hours, at least 94 samples or 92 samples can be tested. So this is a really high throughput one. Besides the manual method, actually this Elasta kit can also be done automatically. So VZM sVNT case is compatible with various type of automatic Elasta workstation to achieve the automatic analysis. With the high throughput workstation, at least 720 specimens can be tested within 2.5 hours. So if we have the analyzer, the process can be reduced very easily. So I can just like click and check. So we just need to prepare all the samples and put them into the analyzer, and we press the start button and wait for the results. So here, all the manual process will simplify to dilute the washing buffer and dilute some regions and pull all the regions into the container and pull the plates and the tubes, test samples into the machine and start the program and waiting for the results. So here is the software, how the analysis operating system looks like. This is the final reports. So after testing, actually we can have the reports like this. So this will show the barcode of the patient's sample and show the OD or the signal of the testing and show the positive or negative at the interpreted results. So how the result interpreted, no matter we use machine or the manually, actually it's all based on this same formula. So for the quality control, the NC or negative OD control should be over 2.8 and the positive control OD should be below 0.2 because it's a competitive method. So if the more neutralizing antibody in the samples, that the signal will be much lower. So if there's no antibodies or the negative sample, the signal will be higher. So based on this inhibition formula, we can get an inhibition number as a different percentage of the inhibition. If the inhibition is over 20%, that means that is a positive one, indicate the presence of the RBD neutralizing antibody is detected. But if the result is below 20%, that means this is a negative sample. The negative sample indicates the neutralizing antibody is absent or below the detection limit. So how about the LOD? We use the monoclonal neutralizing antibody that diluted in the negative serum sample to mimic the positive neutralizing antibody serum samples. And the limit of detection was evaluated as over 92.5 nanogram per milliliter. Then the antibody can be detected by this Elasta kit. So if in the reaction mixture, if the final concentration of the antibody is over 4.6 nanogram per milliliter, then the antibody can be detected. So this method is really sensitive. So how about compared to the live virus neutralization test? So 84 cases of the positive serum from the vaccine subjects with the brain or the live virus analysis titer over 4 was identified by this kit, with the brain 50 titer ranging from 4 to 4 to 24, 24 to 48, and the titer increasing in the brain 50. The median inhibition rate also increased from 5% to 26%, at least to the 75%. So the result of the Elasta analysis is quite correlated to the brain experiments. So here is a summary of the analysis. So the sensitivity of this kit is 96.55 and the specificity is 100% for these 52 samples which identified as negative in the brain experiments. So because as we all know, the gene script have so many publications in to study to compare with the brain 50. And so how about this method compared with the gene script sVNT products? So from here we can see the clinical collected specimens was tested on both gene script Elasta kit and VZM kit. The correlation between these two kits was analyzed and the R-square is 0.294. So actually these two methods is quite correlated. And and also compared to the brain 50. So if the lab cannot achieve the brain 50, I think these methods can be a very good solution to test the neutralizing antibody. So here is the validation we done in Indonesia. So as you can see here, our products have very high sensitivity even at the very early stage of the COVID-19 diseases. So as we see here, we have the total 90% sensitivity in the 108 samples, and the accuracy for 225 sample is 93.8. So except to reduce the qualitative assays, actually this method can also easily achieve the semi-quantitative assays, which means we can give a titer. So to evaluate the titer of neutralizing antibody between samples and compare the efficiency of the vaccines, the semi-quantitative assay is very important and very easily achieved by the sVNT. So the method is just we need to full diluted each samples, and so to check at which dilution fold the negative is transferred from the negative positive sample to the negative sample. The dilution fold for that sample will be as titer. So with the titer data, we can compare the analyzed neutralizing antibody level between different samples. And for so we can also come with the group of the many enough data. We can also compare between group to group between the different vaccine platforms. So we can tell which vaccine have the better efficiency to help us, or after in vaccination, which vaccine can help us to get a high level neutralizing antibody to help us to and protect the fight against the virus. So I think for no matter for the research or the people all around the world, the most concern is how long the neutralizing antibody can last or how long can persist in our bodies. So it is very important to know the antibody, especially the neutralizing antibody kinetics in the circulation system after vaccination. So the sVNT can be used at the very beginning of pre the vaccination, and after vaccination, we can continually test until at which time point the antibody is already dropped to a very low level, and at that time, I think it will be a good time to take the reboost of the vaccination. So as the conclusion, the sVNT Elasta kit is user-friendly and can be easily performed with microplace reader. And compared to the live virus or pseudovirus analysis, the Elasta is time-saving and high-throughput assay, which can be significantly boosted by using an automatic Elasta workstation. And beside the qualitative detection, sVNT can be also used for the semi-quantitative assay, which is an ideal tool to the antibody kinetic assay in the pre and post vaccination study. And this is the brief all the presentation for today, and I'm very thank you for your time to listen, for your attention to let me briefly go through the neutralizing antibody test. Thank you. That's all.